So my name is Ilkhanan Bornstein. I'm an associate professor at the Department of Genome Sciences and organizer of this lecture series. And I want to first welcome you to the last talk this year. And it is my absolutely great pleasure to introduce our speaker tonight, Dr. Tony Blau. So first, just to give you a bit of background, Tony did his bachelor degree at Wright State University and then an MD at Ohio State University. He did then an internship and a residency at Duke University before joining UW, first as a senior fellow and then as a faculty in the Division of Hematology. He's now a professor of medicine at the Division of Hematology, the director of the University of Washington Center for Cancer Innovation, and a co-director of the University of Washington Institute for Stem Cell and Regenerative Medicine. Now, at this point in the introduction, I usually start talking about the speaker's research and his numerous awards, and I can obviously do this for Tony. But instead, I really just want to tell you one thing, and that is that the first time I had the opportunity to talk to Tony, and that was talking about this Center for Cancer Innovation that he was just about to put together, I was absolutely blown away. And I was blown away not just because it was obviously wonderful science, but really because it was very clear that Tony was a visionary. And he was specifically the kind of visionary that has the ability not only to think outside the box, but also have the ability and the talent and the persuasion power to actually move forward with this vision and making it happen. And along the way, bring a lot of people that believe in the same vision and join the journey. And so having said that, I'm absolutely excited to hear Tony's talk tonight. And I'm sure you are too. So please just help me to welcome Dr. Tony Blau for the talk.
Thank you. Oh, yeah, I don't need this. Yeah, thanks so much, Elhanan. That's such a lovely, wonderfully generous introduction. So I'll tell you a little bit about myself first. So I'm a hematologist. That means I'm a blood doctor. I traditionally haven't done that much in cancer, but I'll tell you how I've become, over the last 5 years or so, really kind of engrossed and kind of captivated by the opportunities that are available, I think, in cancer. I think just kind of the nutshell of what I'll tell you is I think we're really at the beginning of a revolution in cancer. I think cancer will be treated 10 years from now in ways that are fundamentally different from the way that it's treated today. But I'm getting to be an old guy, especially looking at most of the people in the room here. That's a reminder of that. But this is what I looked like still when I was much older than a high school kid. But this is really early on when I came to Seattle, and I was learning to do a procedure called bone marrow transplantation. And bone marrow transplantation is this kind of intensive procedure where you're taking a patient with cancer that's fighting for his or her life. Typically, they have leukemia, and you're giving that person, this is how we did it back then in 1993, you're giving that person massive doses of radiation therapy and chemotherapy that essentially is so much treatment that you are condemning them to die from the treatment unless you rescue them with somebody else's bone marrow. And that allows them to live. And the hope is that you give so much treatment that you can wipe out their leukemia, replace their bone marrow with somebody else's bone marrow, and allow them to live to be a little old man or woman. And this was the reason that I came to Seattle, to learn how to do this stuff. This stuff was developed here in Seattle, by and large. And this is a picture now as I'm beginning to be an attending doctor on the bone marrow transplant unit. And this is a picture taken on the occasion of this little girl's 6th birthday. And this picture was taken in the hospital because her mom was in the hospital. And you don't see her mom in the picture because her mom was taking the picture. And what you also can't see then is that her mom, her skin color was literally as orange as a pumpkin. And her mom had developed a complication of bone marrow transplantation called graft versus host disease, and she died just a few weeks after this picture was taken. So as tragic as that is, the real tragedy of this story is that had this little girl's mom been diagnosed with the type of leukemia that she had, called chronic myelogenous leukemia, just a few years later, if this picture was 1997 instead of 1993, her mom would have been treated with a pill that would have almost certainly worked. and her mom would probably be alive today. And so the fundamental lesson is that in the field of cancer, time matters. Matters. In people that are working in cancer research, it matters whether you discover something today or 5 years from now. It matters in terms of people's lives. But traditionally, I haven't, like I say, I haven't worked in the field of cancer so much, but my wife, Sibel, this is a picture of my wife, is a private practice oncologist, and she's really in the trenches. She works down in Puyallup, sees 30 to 40 patients a day, and in talking with Sibel several years ago now, it seemed like the way that we were approaching cancer as a kind of collectively, scientifically, and as a community, that approach probably needed to be rethought. And I'll tell you more about that in a moment. If you look at the fundamental problem that we face, it's this idea of what does it mean to do one's best today. Back in 1993, it meant to give someone incredibly high doses of chemotherapy and radiation. What does it mean today? And if you look, as you do science, one of the things that's very impressive in a science laboratory is how fast things move. The rate of progress is astonishing. And that's represented here by exponential growth. The improvements in technology are breathtaking. And as a scientist, when you go to the lab, it's incredible. But then as a doctor who's seeing patients, when you look at the rate at which those improvements in the lab are being applied to patients, for example, cancer patients, they're improving, but they're improving at a much slower rate, a linear rate of improvement. And so if you're a patient with cancer, you could think, if you're here and you have cancer, you could think that you're facing an ever-widening gap between what's possible technologically and what's actually brought to bear in your clinical care. And I think as researchers and clinicians, our collective goal should be to find a way to close that gap. One of the major advances in technology that's come about in the last now more than a decade has been advances in DNA sequencing. And there's this concept that people talk about a lot, and if you haven't heard of this concept before, it's worth knowing, it's worth remembering, that's called Moore's Law. And that's a phenomenon that's extended back to the 1950s, where the power of computation doubles every 18 months at the same cost. So a computer that you buy 18 months from now will be twice as good as a computer that you buy today, yet it costs the same. And that decline in the cost of computational power has been a long-standing phenomenon, and it's been a real driver of software development and an immense driver of technological advances. And in the field of science, DNA sequencing, the cost of sequencing DNA, which might sound like an arcane thing, but it's actually an incredibly important thing, has fallen initially at a rate that was similar to Moore's Law, but beginning in about 2006, fell at a rate that was way faster than Moore's Law. So this dramatic decline in the cost of DNA sequencing made it possible to think about doing this in patients, not just one individual, but maybe anyone could have their whole genome, all of the DNA in their cells sequenced. And that's opened opportunities that people couldn't have imagined 10 years ago. And if you look at what have we learned from DNA sequencing, what is the fundamental lesson, if you will, from sequencing the DNA of cancer cells? You could ask, okay, what's the fundamentally important lesson that we've learned there? Different people would say different things. What I would say is that the fundamental thing that we've learned is that every cancer is different. And what that means is that every cancer that has ever existed is different from any cancer that has ever existed or ever will exist. And I believe that the field of cancer research is only beginning to come to grips with the profound implications of cancer's uniqueness. And I'll tell you why. The reason why is that the way that we make advances in cancer, and the way that we've done this now for 40 years, is through a process called a randomized clinical trial. And this is how progress in cancer research is made, where let's imagine that this is a woman with breast cancer. And now we want to find a better way of treating breast cancer. How might we do that? Well, let's say we find this woman with breast cancer and we find many other women with breast cancer who seem like they have kind of very similar types of breast cancer. And then we make these two groups, two bins, if you will, and we randomly assign these women to one of two groups. And then we give one group the standard treatment, whatever it is that we give today, and the other group an experimental treatment. And then we say, okay, which treatment worked better for the group on average? And here we show in the boxes, these are the patients that responded in each group. As you can see, the standard treatment worked better for patients on average than the experimental treatment. So what we do then is we say, let's move forward with the standard treatment. Let's discard the experimental treatment. And then we do it for the next round of treatments that we're interested in. But what this strategy ignores is the fact that there are patients who got the standard treatment who didn't respond, who would have responded had they been assigned the experimental treatment, and vice versa. So what this is, so while this approach tells us what works on average better for most patients, it doesn't tell us anything about the features of patients that will allow them to respond to one treatment or the next. In essence, what this approach depends on is that this is a fair comparison, that you can actually divide these patients between two groups, treat them differently, and that it's fair to compare the results, the average results. It's kind of like thinking about how many, I've already told you that genetically breast cancer, every cancer is unique, but if you think about it, this approach is saying, is asking the question, how many different kinds of breast cancer are there? And if you go to the grocery store, QFC, it might be like, how many different types of fruits and vegetables can you buy from the grocery store? And I don't know how many there are. I haven't actually counted, but maybe it's like 3 dozen, maybe 2 dozen to 3 dozen different types of fruits and vegetables. So if you imagine that this represented, each one of these represented a different type of breast cancer, then you could say, maybe if we had a clinical trial that had thousands of patients, thousands of breast cancer patients, maybe each kind of breast cancer would be equally represented between the treatment and control groups. Maybe these differences would even out if there are only 2 dozen kinds of breast cancer. But what if the number of different types of breast cancer is really more like any item that you could buy in Manhattan? And, you get a shoe here or a purse here. There's no number of patients that can accommodate, in a clinical trial, that can accommodate that diversity. And so toward this end, a few years ago, as Elhanan mentioned, we created this thing called the Center for Cancer Innovation. And the idea is, some of the major ideas are shown here. The idea is to first say that cancer is this immensely difficult problem, incredibly complicated problem. And the only way to have a chance of tackling this is to gather people with very different types of expertise. You don't need just a cancer doctor or a nurse. You need really hardcore biologists, really hardcore computational scientists, genome scientists, cell biologists. You need people who barely speak the same language, figuring out how to work together toward a common grand goal. The second idea is that there's no single institution in the world that has all of the expertise that you need to approach this problem with everything that's required. And so the other concept is this concept of transcending institutional boundaries. You want to bring together people in a way that ignores which institution they come from. You don't care where a person works. If they have advice, technology, or expertise that could be helpful to our patients, we want to work with them. The other idea is the idea of viewing cancer as a big data problem. You may have heard of big data. You can get enormous amounts of data from a cancer patient. And big data is all the rage in science. It's used to forecast the next little tick up or down in the stock market. But the idea that people with expertise in big data, the idea is that people with expertise in big data could be valuable to this process of trying to understand cancer. Another idea is this idea of fusing the process of research and clinical care. So traditionally, the world of research and the world of clinical care are separated by this firewall. And this firewall is intended to protect patients from harebrained researchers who want to do crazy things. And so there are all of these protections in place that prevent research from transitioning or that make it difficult for research to transition into clinical care. And what we think is that there are situations where patients with cancer who are fighting for their lives don't benefit from that protection. In fact, they would much more likely benefit if we could, in certain instances, remove that firewall and fuse the processes of research and clinical care so that if there's anything in the world that could help a patient, even conceptually could help a patient, we want to get access to that. And then the other idea is to think, since every cancer is unique, we think we can learn a lot by very deeply monitoring what happens in an individual patient with cancer over the course of his or her disease. So this idea of intensively following a cancer patient over the course of his or her disease is really an important concept for what we're doing, as I'll talk about in a moment. So this idea of bringing together people with different types of expertise, one example of that is a meeting that we had at our house, Isabel and I had at our house. This is the first of these meetings that we had at the end of 2013. And we have called this I think we have to come with a better name maybe, but we call it pizza, beer, that's to draw people in, and then omics, which means genomics, proteomics, and cancer. And so this is, there's a very special group of people that would show up for a meeting like this. It's not everyone by any means, but they're typically nerds. And in this picture, there are people that come from many different types of backgrounds. Computational people. This is Nathan Price from ISB, for example. People in the biotechnology industry, hematology faculty and fellows, clinical doctors, super computational biologist, Brig, Meekum, and even a high school kid who wants to be a doctor, wants to be a cancer researcher. But no single group of experts is sufficient to the task of trying to understand cancer and tackle cancer. You also need patients, cancer patients, who are willing to be the subjects of an unprecedented scientific investigation. And in this picture is Misa Ingalls, who had the type of breast cancer that we decided to work on initially, called metastatic triple-negative breast cancer. That's a very aggressive form of breast cancer. And Misa very sadly died now about a year ago. So just to give a brief overview of the clinical trial, it's shown here. We call it, this is our first clinical trial. It's called the Intensive Trial of Omics in Cancer, or ATOMIC 001 is our first trial. We imagine this type of trial design will be, we can extend that to other types of cancer. But to be eligible for our trial, a patient has to have this type of breast cancer, metastatic triple-negative breast cancer, and she has to have decided that the next drug she's going to get is a chemotherapy drug called cisplatin. So the reason for that is that this is a very aggressive tumor. If you watch it for 2 weeks, you'll watch the tumor grow for 2 weeks. You can't wait to start treating this disease. This disease is incurable. If you get metastatic triple-negative breast cancer, you are very unlikely to survive that. And so what we do then is, before the patient starts cisplatin, we do multiple biopsies. We stick needles in the tumor in multiple sites of the body, and to collect tumor samples from multiple sites. And the reason is that not only is cancer different from one cancer patient to the next, but as cancer begins in our bodies and begins to grow, it's changing. It's changing right before our eyes. And one site of tumor will be different, invariably different from another site of tumor if you look closely enough. And sometimes those differences can be really important. So to try to understand those differences, we biopsy multiple independent bits of tumor. And then we take those little bits of tumor and we do an extensive sequence analysis with these different types of tests that sequence the DNA, the RNA, and then we use a panel. We sequence genes that we know are involved in cancer using this test. And so we sequence multiple independent bits of the tumor. We place it on the cloud, making it accessible to computational biologists across the country. And these are some of the computational biologists that look at our patients' data. And what you end up with is this enormous amount of data. And the challenge is not getting the data. The challenge is, how do you make sense of it? How do you make heads or tails of it? And so the idea is that we find the best people in the world that we can find, and we do our best to put the data together in the best way we know how. We have a meeting of a tumor board and we write a report. And in our tumor board, we say, we think this patient may respond to this drug. So for that patient, we create a prediction or a hypothesis. And then if the patient wishes to follow our recommendation, the patient and her doctor, then we try very hard to make it possible for the patient to get on the drug that's predicted to work. And that sounds easy, but it turns out to be incredibly hard. You can find a patient, you can find a tumor, you can look at the tumor, you can predict that the patient might respond to this drug, but actually getting that patient on that drug turns out to be super hard. If it's an investigational drug, you have very poor prospects. You can try to refer a patient to a clinical trial. That almost never works. It's worked a couple of times for us, but for various reasons that I'd be happy to go into, that almost never works. We've made single clinical trials for our patients. We've made a clinical trial just for that patient. We've done that a few times. You can sometimes get a patient on a drug that's FDA approved. That means their doctor can write a prescription and they can get that drug. But do you know how much a cancer drug costs? It costs, on average, new cancer drugs cost about between $10,000 and $12,000 a month. And so $120,000 a year. And unless the drug is approved by the insurance company for treating breast cancer, the insurance company won't pay. And so getting the patient on even those drugs that are approved can be incredibly difficult, takes a lot of work. But we've been reasonably successful. Through a lot of hard work, we've been reasonably successful at this. So the idea is make a prediction, test it in the patient, rebiopsy the tumor, test the tumor, test how the tumor is responding on a molecular level to these interventions. And over the course of the patient's illness, we do multiple biopsies at multiple time points, and we collect this data. And we think that the information that we will gain from these patients eventually, when we enroll enough patients, will tell us a lot about the rules of how cancers evolve and escape treatment. It'll give us eventually, I think, something that looks like a rule book for cancer. This is just a slide to show that when we do these biopsies, when we stick needles in tumors, we go through a very elaborate process to make sure that we're doing things as well as they can possibly be done. And when you're doing this, this is what it looks like in the radiology suite. When we're doing these biopsies, this is one of our patients. This is an ultrasound machine. These are the people that are actually doing the biopsies. And then all of these people in the back are taking the tissue specimens, looking at them, looking for tumor cell content. This is a microscope where there's a pathologist saying, okay, you're in tumor, you're not in tumor. And so it's a pretty elaborate process. And then we do these different tests where we sequence all the genes, as I've mentioned already. We sequence cancer-associated genes, and we sequence messenger RNA, which tells us which genes are turned on and turned off. And, you know, the field of cancer research right now, the way people tend to use this information is kind of like a Chinese menu, where if we find that you have this type of mutation, you get this type of treatment. So when we look at a patient's tumor, we find dozens, sometimes hundreds of mutations. And one of the first things we do is, oh, do they have one of these? Oh, they have that. Let's try to get that. Let's try to get them on that treatment. So we live still in this linear world where we're looking for this mutation so that we can try to use that treatment. But this is a vastly overly simplistic way. Too simplistic way, way too simplistic way of going after this that doesn't work, surprisingly doesn't work very well at all. And we think we do it better than this for various reasons that aren't worth going into right now. But what really is going on is when your patient has a mutation, has a set of mutations in his or her tumor, is that this mutation or this set of mutations, no single mutation is actually causing their cancer. It's this collaboration between mutated genes, their mutated proteins, and these mutant proteins can interact with each other, but they can also interact with all of the rest of the cell's machinery. And this is obviously a vastly oversimplified depiction of what's going on, but this is a network. And this is a perturbed network where every patient's cancer is setting up this rewiring or this short-circuiting of the cell's network in ways that we don't understand. We don't understand. And we're taking the most primitive, you know, if you think about it, we understand this so poorly that we're taking the state of the art right now is to look at single genes, where what we need to do is we need to develop ways of looking at cancer as a network problem, trying to perturb the network with a drug, and then seeing how the network responds, is altered in response to the treatment. And it's not just that, but also the idea that cancers have cells that aren't just cancer cells, but there are immune cells that infiltrate the tumor. And it turns out that these immune cells, if you were to cite the number one advance in oncology in the last 10 years, it's been the recognition that for many cancer patients, their immune system is trying to eliminate their cancer, to attack and eliminate their cancer. And it doesn't work usually. I mean, obviously, it fails. That's how they got cancer. But by understanding how the immune system is being prevented from eliminating cancer and finding drugs that wake up the immune system has been a hugely important advance. And it's the most exciting advance in cancer in the last 10 years is developing drugs that can allow the body's immune system to destroy a patient's, to have a patient's own immune system destroy their cancer. And that's an immensely exciting development. And we also, when we do DNA sequencing and we do all this other sequencing, we're not just sequencing the tumor cells, we're sequencing the immune cells. And we think we can get a lot of information from that. And we think sometimes that information might be helpful to our patients. So if you were to just summarize what we've done so far in our clinical trial, we've enrolled 12 patients. So these are women who have gone through this process of allowing us to do multiple biopsies at multiple time points over the course of their disease, where the message is, we have two messages for these patients, that we will do everything that we possibly can to try to find something that could be helpful. But I'm not confident. In fact, I'm not certain at all that we can do any better for you than you would achieve by just going to your doctor and getting whatever your doctor's normal treatment is. So all I can promise is effort. We can promise effort like nobody's business. But we know that we're not good at delivering results. We know that. So that's one message. And if you look at the patients who want to participate in this, they're very much attracted to the idea that their disease could have meaning, could allow us to learn things that could help other people in the future. So I think these 12 patients and the patients that follow them are really making a heroic contribution to advancing our knowledge about cancer research, about how cancer behaves. So in these 12 patients, we've done 25 sets of biopsies. We fortunately haven't had any adverse events. This is a big number. We have 6 terabytes of data from our first 12 patients. Our first patient has over a terabyte alone. We've had a number of meetings of these tumor boards. We've generated hypotheses and tested them, and we've gotten these drugs. And overall, we know that that through doing this, we can make this process work. We're quite successful, I would say, at getting the drugs that we want. But we know how hard this problem is and how hard it's going to be to really make dramatic advances. But I think through this process, eventually we'll be able to make significant advances. So just to give you an example of one patient, the second patient in this trial that was enrolled is a 56-year-old woman who had breast cancer initially in the fall of 2007, that was treated and thought to be gone, was in remission. But then in the summer of 2013, it came roaring back. And she had tumor in her bone marrow, and her blood counts were falling because her bone marrow couldn't make blood anymore. And she enrolled in our study. And this is just a schematic depiction of what happened in this patient over the 9 months that she was in our clinical trial. Over that time, we engaged the help of 32 different researchers from 14 different institutions across the world. 20 of these researchers had never touched or seen a patient before. they're in their labs working on, they're experts on some gene, but they've never come close to a patient before. And 18 of these researchers we recruited specifically for this patient. So one of the things that's really interesting about this is that when you find every one of our patients has a laundry list of mutations. You have all of these mutations, and you're just slogging through trying to understand what's important, what might we be able to do something about. And no one can know enough. No one can have the knowledge that you would have to, that has the knowledge that the world has about about what a particular mutation in a particular gene might mean for that patient. But what you can do, for example, in this patient had a mutation in this gene called ROS1. And this was a novel mutation. And what you can do is, you can find out who are the experts in ROS1 around the world. It's easy. And then you just send them emails. You say, we have a patient with a mutation in this gene that you happen to be the world's expert on. Do you think this mutation might be driving our patient's cancer? And do you think that would make our patient susceptible to a chemotherapy drug called crizotinib, which we know can inhibit the function of ROS1? And so we sent emails to a person in Germany, Taiwan, Oregon, and I'll tell you more about that in a moment. But the thing that's encouraging about this is that the researchers always respond. You know, research is a pretty intensely competitive enterprise. I don't know if you guys know about this, but you want to be the first to publish your paper. You want to get it in a high-profile journal. You want to get, and that helps you make your career go forward. You can get grants and things like that. And so researchers are very protective of information that hasn't been published before. If they know something, and if it hasn't been published yet, they don't tend to tell a lot of other people until they've made a splash. But when we send an email to people about this kind of thing, where there's a human being who has cancer, who has a mutation in your gene, no one has told us, we're not going to tell this because we haven't published it yet. They'll tell you whatever they know. If they did and they'll get their postdoc, they'll get their student who's an expert on this to look at these things. So that's been a very heartening development, is that it really is possible to contact experts in a way that where I think there's this untapped resource in the world that is open to being contacted and weighing in on these things. The other thing that we found is that companies seem to be really interested in this. This shows over the course of this patient's illness, there were 4 companies that dedicated a lot of resources to trying to help understand what was going on with her, including a company here in Seattle called RareSite that measured tumor cells in this patient. And this is just a little bit more about the researchers that we contacted. The most interesting story from this was this researcher, Dan Flynn, in Lawrence, Kansas. And I didn't find Dan Flynn. I sent an email to a scientist here at the University of Washington named David Baker. And David said, I don't think we can help you, but he forwarded my email to one of his colleagues, Lance Stewart, here at the University of Washington. Lance Stewart knows Dan Flynn, and he says few people on the planet know more about this question than Dan Flynn. And so Dan Flynn sent a long email saying, he thought maybe this mutation could be relevant. So the idea of creating a social network, in a way, of expertise that could help bring information to bear that could be helpful to your patients, that's part of the idea. This patient happened to have really high levels of tumor cells that were circulating in the blood. So breast cancer cells that are circulating in the blood, really high levels. And we used those to monitor what was happening over the course of her treatment. This is initially the cisplatin treatment. And then at some point, she couldn't take that anymore. Her circulating tumor cells rose. And she was started on this drug to try to hit this mutant gene called ROS1, this crizotinib that I mentioned. And that was a prediction by the smartest minds that we could find in the world. And what this shows is that her circulating tumor cells went up. They continued to go up on crizotinib. So this is a prediction made by the best people that we could find in the world that didn't work, that failed. And she died a couple of months after this. This is what some of the circulating tumor cells look like. These green cells are circulating breast cancer cells that clumped, and in the days prior to her death, clumped in the lungs, and these are circulating tumor cells that landed in the arterial system of her lungs and were the cause of her death. So this idea of trying to gather the world's experts to try to help our patients led to this concept of something that we call the tumor crowd modeling platform, where we now have begun to place our patients' data on this open access web platform that will allow anyone in the world to comment on their tumor. And I won't go into it now because of lack of time, but we're undergoing a massive expansion of this, where initially we had 2 patients from our clinical trial that were here, where experts could comment, experts could see each other's comments. But now we're going to expand this to any patient with cancer. And so any patient with cancer who wants to make his or her public, de-identified clinical information and molecular test results available for global expert comment, anyone in the world to comment on. And we're doing this, we're launching this initially in collaboration with my wife's practice, Sibel's practice, where we're now, the latest is that we have 200 patients that over the last month that have signed up for this. And, but we think that this can scale to millions of patients because we've created a way to, in an automated way, to pull information from the patient's electronic medical record to make these summaries automatable and scalable. And so the way I used to end this talk was to say that what this would allow someone to do is to take, imagine 1 million patients whose molecular information, clinical treatments, and outcomes were available longitudinally over the course of their disease, were available on this type of platform. Then what I used to say was that if I got cancer someday, that would allow me to say, whose tumor does mine most closely resemble? Who among those 1 million patients do I most closely match? What treatment did they get? What worked for them and what didn't? And then have my own, I'd like to find out which treatment worked for them and let me get it. But then let my own experience add to that ever-growing body of knowledge. Ultimately with the idea of every cancer patient's experience adding to an ever-growing body of knowledge that makes us progressively smarter about how cancer works. Today, patients get cancer, they get treated, they respond or they don't, and collectively we learn nothing from the experience. So the idea is to move toward this time where every cancer patient's experience makes us smarter. And so this is a picture of my doctor, Sam Warren, who's in the audience. I'll just tell you a few things about how this story now ends differently from the way it ended a few months ago. Because I saw Sam at the beginning of this year with some hip pain. I'd walk around, I'd get some, and it didn't feel like traditional hip pain. I thought, oh, I'm just getting old, my hip's worn, I'm going to have to get my hip replaced. But it felt like a hip pointer, like a football injury where you get hit in the helmet in the pelvis, and it's kind of sore. And it wasn't going away, and Sam ordered an X-ray, and it hadn't occurred to me that there would be anything seriously wrong until the moment that the X-ray machine beeped. And I thought, you know what? This does seem a little weird. I better take a close look at this X-ray. And it popped up on the screen instantly, just as soon as I got it. And initially, I'm looking at my hips, and I'm thinking, those look okay, no big deal. But then I'm a hematologist, I'm not an x-ray guy, but the other thing that I remember you're supposed to do is you're supposed to compare the symmetry of the two sides. Does this look the same as this? And you can't see this very well, but this is a hole. It's this mean thing eating my bone. And you can see it's right at the edge here. It's starting, and so within 5 seconds of getting this x-ray, I knew I had cancer. I knew it before the x-ray technician, before the radiologist saw it. And so that was a surprise, that's for sure. I called Sam. He helped me get the workups. Sibel helped me get a couple of things done really quickly. The next this is a PET CT scan that showed this big thing in my pelvis. The next day, that was done the next day. And Sibel's partner, Andrea Veach, a lovely friend, just wonderful friend, did a bone marrow biopsy on me the following day. This is on a Friday. And by the end of Friday, we knew that I had this disease called multiple myeloma. And so multiple myeloma is a disease of the blood, a kind of cell called a plasma cell. And it's up until now been considered largely incurable. People have traditionally thought of it as being incurable. But there's a but, and I'll tell you about that. The reason that most people know about this is because Tom Brokaw has multiple myeloma, and he wrote a book about it, and there's a special. And the thing that I learned about this is he's a fast writer because he got this disease just at the end of 2013, and he already has a book about it. But it's been interesting. So on Friday, April 10th, I knew that Sibel and I were driving back from Tacoma, and she called our friend, who's a wonderful hematologist here, Pam Becker. Her lab's actually one floor below mine, and this is her husband, Mark Stewart, who's a medical director at the SCCA, at the Seattle Cancer Care Alliance. And that night, they came to our house. They brought a Boston cream pie, I don't know why, and a bottle of wine. And Pam, told us, so going from Thursday night, knowing you have cancer, you don't know what kind, you don't know whether you're going to live for 3 months, or myeloma was about as good as it was going to get, I think. To having a diagnosis, and your doctor there the next night was stunning, obviously. But then Pam, so Pam's my cancer doctor, and so what I had decided to do, I have a colleague that kind of went through a very similar thing, had a very similar kind of idea about cancer, approach to cancer. He's a neurosurgeon at Swedish Hospital. His name's Greg Foltz, and he developed a fatal form of cancer, pancreatic cancer, and he didn't tell anyone. Because he was concerned that it would jeopardize his career. I decided to tell everyone, and I tried to make myself the poster child for what we're doing. Make myself kind of do everything that we've been doing for our breast cancer patients to me. And in a way, I feel like there's no one better prepared for this than me. The network that we've developed and assembled for breast cancer now, we're turning toward me. Beginning with, this is me in the same suite that I showed our breast cancer patients, Pam sticking a needle in my butt. And then so we did a lot of biopsies of that mass in my pelvis. Pam did a bone marrow biopsy. We sequenced all of the different pieces. We collected 300 gigabytes of information with exome sequencing and RNA sequencing. Not that this is going to make any sort of immediate finding that's going to make the determination of what should happen with me, but the idea is to try to characterize this as carefully as we can for the purposes of imagining that this information might be helpful in the future. And what we found, essentially, kind of the most important things that we found is that that big thing in my pelvis was a big collection of myeloma cells called plasma cells. And we compared the myeloma cells in my pelvis to the myeloma cells that were scattered elsewhere in my bone marrow. And we saw a difference in the myeloma cells in my pelvis in that they had a feature, a molecular feature that told us on a molecular level they're more aggressive. Those are the ones to worry about. And whereas the myeloma cells that were otherwise scattered throughout my bone marrow lacked that feature. So we want to keep pretty close track of that. Just kind of the great thing, the really fortunate thing, is that there are a lot of treatments. Unlike metastatic triple-negative breast cancer, where the options are terrible, in myeloma there are a lot of great options. And the initial treatment that people start on, that I have been on now, is this combination of 3 drugs. And I have a kind of myeloma called an IgA myeloma that's responded remarkably well to this treatment. So I'm lucky. But this treatment works really well for most patients with myeloma. I'm very lucky to be responding like this, but I'm not unusual for myeloma patients. And this is a PET CT, the same one that I showed you before in April, and now this big thing has gone down to something that's very small. I asked them to re-biopsy that. They stuck needles in that again, and they had a very hard, there were a few tumor cells there, but they had a really hard time finding tumor cells. The other thing about myeloma is that there are lots of new treatments. So this is a disease where 20 years ago, this really was a fatal disease. There were very few, the prospects were incredibly grim. But this is one of the types of cancer where the rate of progress is probably better than almost anything else, almost any other area of oncology. The advances in myeloma are really impressive. So if one were to choose a type of cancer where you wanted to ride the crest of progress in oncology, myeloma is a good choice. So there are all these drugs that are becoming available, that are available, and it's kind of this pipeline of new things that make me very optimistic, very hopeful that I could live many, many years still. And if I were to talk about this realistically, I know that I could die from this, but I don't think I'm going to die from this. But the thing that occurs to me, so this is kind of where I show the rate of progress. Improvements in myeloma treatment are going up. But the other curve that I think about a lot is that my disease is going to become more resistant over time. So as the cancer is growing, as I mentioned to you, it's constantly evolving. There are slight differences between the different bits of tumor. And after you give a treatment, there are likely to be tumor cells that are resistant to that treatment. And when they grow again, it's likely that their progeny, the daughter tumor cells, will be more resistant to treatment. So this is just a fact of cancer. It's evolution in the lifespan of an individual, where and evolution's smart, and you have to try to find a way to combat that. And it makes it very appealing to think of ways of trying to really hit it hard at an early time point in the course of the disease. And then I thought I'd just finish up with a couple of surprising things that I've learned now. It's remarkable to me. I thought, we've worked in cancer, thought about it, eaten, drank, slept cancer for many years. What more could there be to learn? But you find out that as a cancer patient, there are actually a lot of things that you could learn. The first is that it is, and this is just a couple of things that I haven't thought about it a ton, but these are some things. The first, so this is like my pills in the morning. I go from 0 pills every day to something like, 30 pills a day. And it is so much harder to do than I had ever imagined. It's so much harder in 2 ways. The first is that that everyone thought that I should take 3 drugs: Revlimid, Velcade, and dexamethasone. Revlimid costs about $120,000 a year. Velcade costs about $120,000 a year. And dexamethasone costs about $50 a year. So the insurance company, our insurance company has a policy that you can have Velcade, one of the two, $120,000 a year plus Decadron. That they won't argue about paying for the dexamethasone. You can have one plus dexamethasone, but you can't have both, because there hasn't been a randomized, double-blind, placebo-controlled trial that's shown a difference between having all 3 and having only 2 of the 3. Whereas, as a guy that's thought a lot about cancer and knowing how the cancer is going to evolve over the course of treatment, that just was not a desirable course. So Sibel, through a lot of discussions with the insurance company and with higher level people at the insurance company, was able to convince them to give us these 3 drugs for me. But I never really understood. I'm a beneficiary of a connection that a normal cancer patient would never benefit from. The second thing is, once you have the pills, just taking them right is so much harder than I would have thought. I used to think, patient screws up their medicine. I used to think, come on, man, why can't you get this right? But being on the patient side of it, it is much, much harder than I would have imagined. And then another thing that I would not have thought, but has turned out to be really useful for me, is seeing this fellow, Dan Labriola, who's a naturopathic doctor. And I can tell you that I would have been the least likely guy to see a naturopathic doctor. But one of the complications of bortezomib, one of the chemotherapy medicines that I'm taking, which, by the way, is easy to take. It's a shot twice a week. It doesn't make you feel sick. That's another super fortunate thing here, is you can take all 3 drugs and you work full time, you feel good most of the time. But the Velcade, the bortezomib, can cause something called neuropathy, where you get tingling of your fingers, and that can get progressively worse. And so, and it turns out that there are certain differences between people, genetic differences, that can tell you whether or not you're at a higher risk for getting this neuropathy than average. Are you at an above average risk for getting this neuropathy or not? So after about the second cycle, so-called cycle of Velcade, I might be imagining things, but I'm thinking, boy, I think I'm getting a little bit of tingling in my fingers. and by that time, I had my genome sequenced, and my doctor, Pam Becker, had written an article talking about which genetic changes place you at an increased risk of getting this neuropathy. And so we looked at my genetic sequence, and I had an increased risk. I had a couple of genetic changes which really increased my risk for getting this complication. And I had given a talk at Swedish Hospital where Dan's a friend. I've known him for a long time. He was at my talk at Swedish Hospital. I told him I had myeloma. He said, well, with Velcade, Velcade neuropathy, there are a couple of things that I can give you that will make that go away. That'll just take care of it. And initially, I didn't want to take it because I didn't want to take anything that could in any way counteract the benefits of the chemotherapy. But by the time that I was getting these little bit of side effects, and I knew I had a genetic change that placed me at an increased risk of that, so I went to see Dan. And it turns out to be, simple things like glutamine, an amino acid, and B6, a vitamin. And I took these, and my neuropathy went away. So again, it could be in my head, but it allowed me to take 2 more rounds of this Velcade without any more problems. And then the other really incredibly fortunate thing for me is to have a wonderful, wonderful family. And the other thing that I've learned is in talking with each of them, but then in a discussion that I had with our 10-year-old Melissa. Melissa really wanted to know, is there any chance I could die from this? And I told her, yeah, and I can't promise you that I won't die from this, but I don't think I'll die from this. And for her to know that, and then she initially said, well, I think I'm going to tell one or two friends at school about this, but I'm not going to tell anybody else. And I said, well, you know, hon, if you tell one or two friends, they're probably going to tell other people, so you might want to just tell everybody. But I find, you know, one of the other incredibly fortunate things for me is to have such a wonderful family that has really just been drawn closer by all of this. So I'm an immensely fortunate person. I think this having happened to me just emphasizes in my mind, kind of heightens this sense of urgency that we need to get this approach to treating cancer on stable footing so that it can continue for many years to come. And that's it. Thanks very much for coming.